So good morning, everyone. Thank you so much for tuning in to our My Field for Dummies webinar series. Just a little bit of housekeeping. We are going to be taking a break for December, so there's not going to be any webinars for next month. However, if you tune in for January, we are going to be trying to plan things that are a bit more workshoppy. So we might actually have kind of some new series is going on moving into the future. So do stay tuned on the GBATnet Facebook page and website to be up to date on all of the online events that we are hosting. So for today, I am very excited to introduce the very brilliant Dr. Lydia Nixon, who is a visiting professor over at North Dakota State University, where she also completed her PhD work. And she covers a field that I am personally very interested in, having worked on a portion of this for my own PhD work as well, which is endocrinology and its application to conservation and specifically bat conservation. So without further ado, please take it away, Lydia.
Great. Thank you so much. I'll just share my screen. Okay, hopefully that looks good on your end. Okay, great. And I'm also just going to turn off my camera. Okay, perfect. So, thank you so much for that introduction. I'm really excited to be speaking with everybody here today. Um, I'm going to start with just going into why conservation endocrinology and conservation biology in general is so important. So as we know, the world is always changing. Unfortunately, it's also changing in some big ways, largely due to human activities. We can see the effect of these changes in terms of habitat loss, deforestation, overexploitation of different species, as well as pollution and, of course, climate change. And so the goal of conservation biology, which is the first thing I'm going to talk about, sort of this big broad field, is to determine natural and human-driven impacts on wildlife populations. And this research will hopefully provide answers to specific questions that we can apply to management decisions. So studies have been done on roadkill, the effects of animals being hit by cars, animals trying to cross roadways. And one of the ways that we can manage that is through green bridges, like this one in the picture on top, where the animals have a safe way to cross the road. Another big goal of conservation biology is protecting and restoring biodiversity. And these goals are accomplished using various scientific methods and techniques, one of which we'll get to is the conservation endocrinology. So an example of, excuse me, sorry about that. There we go. An example of a success story in conservation biology would be the reintroduction of wolves to Yellowstone. Wolves were eradicated from Yellowstone in the 1920s, and because of this, there was an ecosystem collapse in this national park, otherwise known as a trophic cascade. What happened was because the wolves were no longer present, the elk populations had no natural predators, so the elk populations just exploded, and this resulted in severe overgrazing of willow and aspen trees. Coyotes became the apex predator of the area, and so their populations also exploded, which decimated populations of pronghorn antelopes, drove down other populations as well, like red foxes, rodents, birds. And then the beavers also virtually disappeared from Yellowstone National Park because of the tree loss, those willows and aspens. And this in turn affected not just plant and animal species that relied on the environments created by beavers, but also a lot of the abiotic factors in the environment, like the actual health of streams and rivers. So wolves were returned to the ecosystem starting in 1995, and it's been really, really beneficial at reversing these issues. Beaver colonies have increased. All of those plant and animal species that they affect have also increased. Elk numbers are reducing since now they have a predator. And so again, this is an example of the power of successful conservation biology intervention. In the last couple of decades, there's been more attention on organismal biology of individual species for conservation biology. So, taking individual species responses to changes in the environment, to influences of human disturbance, and then using that research to scale it up to ecosystems. And this can help us to predict population declines, hopefully before they start. So I included this logo here from a university lab because I really like the tagline, taking physiology to the field. And so we're going to start focusing on the endocrine system now and endocrinology. I'm just going to kind of piggyback off of their logo there and say we're going to be taking endocrinology to the field. Before we do that, though, we're going to just do a brief introduction to the endocrine system. So the endocrine system, in its basic definition, is a chemical bioregulation system that's composed of specialized tissues. These are the glands and the organs associated with the endocrine system, and the hormones that they produce and release into the body. The endocrine system helps to coordinate the development of all of these other systems that we're seeing in this picture, and also how they function throughout an animal's entire life. The endocrine system is also responsible for helping us get back towards homeostasis if something knocks things out of balance in our bodies or in our environment, helps animals respond to change, and mediates the immune response. So even things like resisting illness or overcoming an illness, that's mediated by the endocrine system. So it's a really, really vital system in the body, and it affects absolutely everything going on within an organism's body. Furthermore, the endocrine system is highly conserved. So many parts of the endocrine system are conserved across species and taxa over hundreds of millions of years, 5, 6, 700 million years. So the structures and the hormones that we're studying when we study endocrinology are very similar across many taxa, even evolutionarily very distant taxa. In a nutshell, here's an understanding of the endocrine system. An organism will be perceiving an environmental stimulus. In the picture here, we can see that these are all outside stimuli. So things like weather change or pollution or a dam that's changed their physical environment. It could also be an internal environmental stimulus, like a fever or an infection. So this environmental stimulus is perceived by the organism, and then there is an endocrine response. So this is where we get all the complexities of the actual cascading endocrine response throughout the organism's body, which eventually results in physiological and or behavioral change. So I have an example of that here with a cow. And so we can see with the number 1 on the bottom left that there is a stimulus being put on the mother cow. And the stimulus in this case is her offspring suckling. There's receptors in the udders that can sense that suckling, and they send that information up to the brain, to the hypothalamus. The hypothalamus is the master gland or the control center of the endocrine system. And this will trigger a cascade where hormones, you can see this cascade starting with number 3, 4, ending at number 5, where hormones are released. Those are the green dots in the pictures, with the goal of getting to the target tissues, in this case, the mammary gland, and triggering the final response, the goal of this whole system, which in this case is the desired outcome of milk let down to the offspring. And so this is, again, sort of in a nutshell, one example of how this system works. And it's an example that's focused on a physical response, the milk letdown, but hormones can also affect behavior. So it's important to keep that in mind. So now that we have that introduction, we're going to take a look at the endocrine system within the realm of conservation. And this leads us to conservation endocrinology. This is often referred to as a subdiscipline or a subfield of conservation biology. And it's basically what we've been talking about, the study of the endocrine system with the goal of using those data, those studies to conserve endangered or threatened species. And so it's a two-part kind of work where first you're determining how the hormones that you're studying are influential for animal survival, growth, reproduction, and fitness within the context of their environment. And we have to keep in mind that these environments are always changing, especially in certain very rapid ways due to human influence. And then with this knowledge, we can hopefully implement endocrine monitoring into our intervention or our management that we're doing to protect species. So in these two pictures, we have the example of salmon on the left swimming upstream, and this would be during their mating time, spawning time, and then they would be swimming downstream after the spawning event has taken place. We can monitor the hormones of these fish and to see how pollution in the different rivers and streams are affecting them, affecting their physiology, affecting their reproductive success. On the right with the lions, there's a recent study that showed that the reintroduction of animals, which usually doesn't go so great, it depends on the species, it depends on the environment, but when you're trying to use a method of breeding in captivity for reintroduction in the wild, a lot of times those animals that are released into the wild don't do very well. And so it has been shown with these lions specifically that if you introduce oxytocin to the lions that you're releasing and the lions already present in that environment, that can benefit the reintroduction process and allow for acceptance of the new individuals by the established community. And oxytocin is colloquially referred to as the love hormone. So you can see how it could make the animals be more accepting of each other. And those are just a couple of examples of potential benefits of endocrine research in conservation. So typically we see endocrine disrupting compounds being studied very heavily in terms of endocrinology and conservation. We also see it being used in reproductive monitoring in captivity or captive breeding populations. Also in reintroduction efforts. So what I was just describing with the lions or with the black-footed ferrets, like in this picture, you see a lot of endocrine monitoring in these situations. And then finally, the last way that we typically see the endocrinology research being used in conservation is with population control. So in this example here with the deer, they're working on lowering deer populations using effective birth control, essentially. So those are the main ways that we currently see endocrinology being used in conservation, but there's a lot of room to make it better, to go different routes with this type of research and this huge system. So very briefly, we can collect a lot of different parts of animals or excrement of animals to actually analyze for hormone levels. We have lots of examples here. There's urine, feces, scales of fish, blood samples, feathers or fur. And we'll talk about these with bats later on, specifically which types of samples we take. But there's a wide range of different things that can be studied with regards to actually quantifying the hormones of interest. So now we're going to switch gears again, and we're going to focus more on stress. We're getting into the stress physiology part of the talk now because, as you might have taken from the title, this is where I currently focus most of my research. So this is going to be the main focus for the rest of the talk. So when we talk about stress in terms of endocrinology, we're talking about this stress response system, and you might have heard it just casually called the fight or flight response or something similar, and it's intimately connected with health and fitness. High levels of stress, especially high levels of stress that persist over months and months, are often linked with negative health impacts. It's not always the case, but we often see that. The problem or the issue with that potentially is that the environment is full of stressors. So animals have to be able to adapt and overcome these stressors in order to survive. When we're talking about stress physiology and hormones, the ones that we're specifically focusing on are the glucocorticoids. These are a class of steroid hormone, commonly called the stress hormones. And the two major ones are cortisol or cort and corticosterone. When a stressful event is perceived, that's going to go to the hypothalamus, similarly to how we saw the suckling being perceived by the mechanoreceptors and communicated to the hypothalamus. So the hypothalamus receives this information that there is a stressor on this organism, and it's going to communicate with the pituitary gland using a hormone that then communicates with the adrenal glands. Those are right near the kidneys, using a different hormone. This is part of that cascade. And then the final step of this axis here is when the adrenal glands release cortisol or corticosterone. Usually with an animal, you see one being the dominant type of hormone that's released. And this actually goes to the target cells and will affect some sort of change to deal with that stress. As I mentioned earlier, this is a highly conserved system. So it is a very similar or the same system across many taxa, including bats. So the same system we see in humans, we see in bats. So now a quick look at what glucocorticoids actually do. There is a lot that they do. This is a very truncated list of their functions, but one of their big ones, of course, is mediating that stress response. They also help in maintaining digestion and metabolism, the immune response, and inflammatory responses. And it's important to keep in mind that there's always some level of glucocorticoids circulating in the body. This is necessary to maintain healthy cell functioning, and this level can change within a range. You can sort of see in my graphic here, there's like that green area where it might fluctuate a little bit depending on an organism's needs at a certain time of day or a certain season. But there's a range of circulating glucocorticoids that's necessary and healthy. And again, this is going to allow us to respond to stressors. So if a negative stressor is perceived, the body needs to do something to combat that. Maybe we need to mobilize our muscles so we can run away from a predator. Maybe we need to amp up our immune system to fight off an infection. We need to have something happen in order for us to respond properly to that stressor and be able to survive it. So that's what we see here, where all of a sudden there's this stressor that's been perceived, and the stress response system has to go into an overdrive state to produce and release more of those glucocorticoid hormones. If the stressor that's perceived is acute, meaning it's only a short, existing over a short time period, like there's a predator, we're going to run away, and then in a few minutes we'll be in a safe space, that's known as an acute stressor. And what happens after the acute stress event is over, everything goes back to that basal level that helps us maintain and function normally. However, we also mentioned earlier the chronic stressors, and these are stressors that can last days or weeks, even years. And if you can imagine what's happening in this graphic here, if that was going on in an organism's system for years or even just months, that can be incredibly damaging. It can be damaging to the target cells. It can be damaging to the entire stress response axis. And then when it's needed next, it might not be able to actually perform. So this is where we start to see those negative consequences that affect health, fitness, being able to reproduce, have healthy offspring. This is where problems can arise. So when we want to use glucocorticoids in conservation research, we're really trying to address this problem in conservation, which is the post hoc nature of connecting disturbances in the environment with population numbers. And so what we typically see, especially with glucocorticoids, is that we're already approaching populations that are struggling. We've identified that these populations have had declines in numbers. or the animals are unhealthy, or they're not having offspring that survive. The population is already struggling, and we want to take corrective action. So we can monitor their stress levels using glucocorticoids to see how effective our treatment is. And that's great, but what if there was a better way? And this is what really interests me in terms of the future of stress hormone research in conservation biology. So this better way would be to try and measure glucocorticoid levels to identify those struggling populations before there are population declines. If we can use the stress hormones of these animals to say, uh-oh, they're in trouble, but we haven't seen population declines yet. We haven't seen reproductive declines yet, and things like that. Hopefully we can mitigate whatever is a negative stressor on those animals before the population declines start. And so that looks like this. We have an at-risk population. We think something might happen to this population in the future based on pollution or climate change trends that we've seen. And we're going to start measuring their glucocorticoid levels now. This will help us confirm that the population is struggling. So if it is, we can start taking corrective action and continue that purple cascade to monitor the glucocorticoid levels to see how effective our intervention is. If it's not struggling at the moment in terms of what we see from the glucocorticoid measures, we can continue to monitor them. So we don't just ignore these populations, but we can continue monitoring them, try to implement some management strategies. Hopefully they never get to the yes box. We can improve their situation before they actually become a struggling population that's declining in numbers. And so another way to look at it is that we typically determine struggling populations or species using some physiological metrics like reproductive success, changes in migration patterns, looking at body condition, like with these elephants, and or inferences that we've made regarding ecological or environmental changes around the animals or the populations that could potentially mean that those animals are struggling, so like climate change. But we can also add glucocorticoid monitoring to this work in order to identify those populations that are experiencing chronic or severe stressors and are at risk, but again, they have not yet experienced decline. So kind of getting ahead of the curve. And this would be employing the court fitness hypothesis, which basically says that if an animal or a population is exposed to an environmental challenge, some negative stressor, there's going to be elevated glucocorticoid levels over a long period of time. And with those elevated levels of glucocorticoids, you see negative health and fitness effects. So when we specifically bring this to bats again, we can collect a number of different sample types to evaluate when we're studying glucocorticoids. So we can take blood samples, we can collect guano, the fresher the better. We can also collect fur or wing and tissue punches, or urine. I don't have a picture of that up there, but we could also collect urine. And all of these are adequate ways of assessing an animal's stress levels. The benefit of blood is that it shows the animal's circulating glucocorticoid levels in the moment when you collected the blood. So you're getting a snapshot of their exact stress levels at that moment of capture, but it does change really quickly. So you have to get that sample very fast. The others show a more longitudinal look at the glucocorticoid levels because they're deposited or their metabolites are deposited in the skin, in the fur, in the guano over a period of hours or days or even weeks. And so you can track long-term how the glucocorticoid levels have been changing. So there's benefits to both, but it depends on how you design your study. So I'll show you under the blood picture is going to disappear, and I'll just show you a video of a venipuncture process for collecting blood from a bat. So a little content warning here, there will be blood and needles. I also want to apologize for the quality of the video. I was taking it on my cell phone while also getting the sample. So it's not exactly the greatest quality video, but This is how I would collect blood samples from bats in the field. And that's all there is to it. The larger the bat, the easier it is, of course, because you'll have larger vessels. But it's a quick venipuncture for getting a blood sample that can then be spun down in the centrifuge to remove the plasma. And the plasma is where the glucocorticoids are actually can actually be found. So currently, when we talk about bats and endocrinology, we don't hear too much about questions of conservation. Typically, what's being researched are questions related to reproduction, digestion, or winter torpor, including fat deposition and survival through the winter. And while these are all very important areas of research, it does open us up to this question of, but what about using endocrinology for bat conservation? Are we doing that? What are we doing? And honestly, this is a huge gap in bat conservation research that I believe requires a lot more effort. Of the limited studies available on this topic or relating to this topic, most of the bats that have been studied in this regard are tropical bats. And the limited studies that are out there largely are concerned with baseline levels of hormones, of these glucocorticoids. And those are really valuable, but they don't directly try and answer a conservation question. So I have a couple of examples here. The first is from Spain, where they were looking at cortisol levels in guano from bats that are living in bridges and seeing it was a longitudinal study about fitness and reproductive success. And then another study from Texas was comparing the baseline cortisol levels of bats in bridges versus bats in caves. So again, we can see that there is a conservation aspect to both of these, but it's largely using baseline analysis and not getting at questions that are, I would say at least, rigorous enough to be used by management professionals to try and help conserve bat populations. So there's a lot more that we can do here. There are some limitations that we should be aware of when we're talking about glucocorticoids or other hormones in conservation. And so for glucocorticoids, using them as a proxy, a direct sort of one-for-one proxy for individual or group fitness, that's still largely theoretical. And part of this is because you can't just look at the glucocorticoids. You have to look at them within the entire system of the animal, and it's a very complex thing. So our studies have to be really rigorous and really well designed. One of the concerns here is habituation to repeated stressors. So if we think about that study in Texas, the bats living in the bridges were hypothesized to be more stressed out, experiencing more negative stressors than the bats living in caves, because the ones in bridges have things like cars, noise, car exhaust, all of these things that we would perceive as negative stressors happening to them on a daily basis. And what they actually found was that those bats, at least throughout most of the paper, they showed that they had lower levels of circulating glucocorticoids, which would seem to indicate they're less stressed out. And so it could be an example of habituation, but habituation is really hard to define and to actually observe or quantify. Another issue is receptor density or receptor sensitivity. Again, this is a limitation that we have to take into account, along with which stressors are actually perceived as negative by these animals. Do we know, if they are stressed out by something that we would think is negative? the car noise in a bridge. Maybe they find that soothing. I would find that probably pretty annoying and stressful, but maybe it's not stressful to these animals. And so we can't anthropomorphize what they're experiencing and just assume that it's a negative stressor. So that's another issue with glucocorticoids in conservation research. However, glucocorticoid requirements, understanding these, it's really important for health maintenance of these animals, and it will provide critical information for species and ecosystem conservation. It is a very important avenue of study that can have a really big positive impact for our conservation of bats, especially when we consider things like climate change and anthropogenic influences that are having massive, wide scale, and very rapid negative effects on these animals. So, of course, more research is needed, but we have to be very thoughtful and rigorous in our research approaches, especially because of the limitations that I've already established that we need to take into account whenever designing a project and implementing a project. So, for example, I recommend integrating laboratory and field approaches, not just relying on captive bats, but trying to do the same research in bats in the wild. There's very little research being done in this area on wild bats. It's mostly captive bats. So it's really important to have the field approaches also integrated. Also trying to increase our understanding and the diagnosis of habituation. So again, we can consider that in analysis of our data, and we're not drawing conclusions that are inappropriate. And then also increasing species level research. So a lot of hormone, the way that the hormonal systems in the body works are species specific. So even though we can try and draw conclusions based on other bat species research, there are going to be differences at the species level. So increasing that is really important for helping individual species. And that is where I will leave it. As you can see, there is a lot to do, a lot that can be done with this wonderfully broad and as of yet understudied area. So my take-home messages for you today are that bats, of course, are necessary and wonderful animals. They help us maintain healthy ecosystems, and they're economically important, of course. They're also massively under threat from all sorts of influences like disease, habitat destruction, climate change, and increased persecution since COVID, as we've seen. People's perceptions of bats are definitely not improving overall. Glucocorticoids and all hormones are vital for health and fitness, but they are, as of right now, understudied and poorly understood for most bat species. Again, huge gap in the data out there right now that we can start to work towards filling. And there's also a really great potential for new management strategies for bat conservation. We are in desperate need of those, of enhancing our management practices and toolbox for bat conservation. So this would be a great avenue of potentially formulating some new management approaches for bats. And in addition, this will enhance the efficiency of our conservation efforts. So with that, we have a little bit more time than I thought we would, which is great. If anybody has any questions, I'll be happy to field those. Thank you so much for tuning in.
Yeah. Okay. All right. Thank you so much, Lydia. That was an incredible talk. We did get a good question in the chat. So going back to actually taking samples from bats, how do you ensure that the bleeding stops from when you are taking a sample from a bat?
Sure. So the first really important thing is to make sure that you're only puncturing veins and not arteries, because they have a much lower pressure than the arteries. So only target veins. And then you might have seen it at the end of the video, but again, the quality was not great there. You would apply gentle pressure, steady gentle pressure with clean cotton on each side of where you made the puncture for about 30 seconds. typically that's enough to stop the bleeding. If it doesn't stop, you would always carry styptic powder with you, and you can dab some of that on there, reapply that gentle pressure for another 30 seconds or so, and that should do it. Um, Also, you can keep the bat for a little bit afterwards if you want in an individual cloth bag, maybe for 10, 15 minutes, just to make sure that it doesn't reopen. But I've never seen that fail. And that's a pretty standard approach to making sure that that bleeding stops.
All right. Do we have any more questions for Lydia? You can always put them in the chat or you can chime in uh, with your mic. Oh, they say thank you.
Thank you. Also, feel free to email me. This will, of course, be up on the YouTube page, so you can find my email there if you have questions.
Mhm. Yes, and as we wait for any questions to come in, I will say again that this talk, if you want to go back to it, it will be up on the YouTube channel probably within the next day or so, or we will have the PDF available on the GBATnet website as well. So we can always go back. All right. Well, if we don't have any more questions, we did get a nice little group in here this morning to join us to talk about endocrinology. So again, oh wait, we did have one more quick question. I don't know if you can see the chat or not, Lydia. It says about validating the stress hormone assays for bats. Do you have any recommendations on what kind of antibodies or kits to select?
So I use a commercially available assay kit through Arbor Assays. I've never actually made my own antibodies. So I unfortunately don't have too much advice on that, except to find someone that has done it. So if somebody's published a paper where they said they designed their own, rabbit antibodies that they were then using, reach out to them for methodology and help. But I ordered a couple of different kits and then tried them and saw if my response curves were the same as theirs. And that was one way to validate the kits to make sure that they were appropriately reading what we would expect. So that's one way to do it. I also asked the companies, since we were trying them for a new species, if we could either have them for free or at a discount so you can get those multiple kits a little bit easier. That's the most expensive part of this type of analysis is the antibodies and the other solvents and things that go into those kits. So that's the way that I found was the best way to approach it, was to get multiple and then see the curves, the response curves of my measurements for my samples versus their standards and see if they match up. And maybe Dana has some other advice there too, since she's done similar research.
That sounds about right to me. We did have another question in the chat as well. Has anyone used wing punches to quantify cortisol? It was listed, but I was curious if it's been done.
To my knowledge, it has not been done. Perhaps there is something in the literature that I'm overlooking or not remembering at the moment. But not to my knowledge. I have seen it with other hormones, but not with cortisol or corticosterone.
I also second that. I've never read it in the literature before. But if anyone else has heard of anything using wing punches rather than other types of tissues or fluids, then, then let us know. All right. Okay, well, if we have no other questions, I'll give it a second because we did have a couple of questions come in. All right. Okay. Well, everyone, thank you so much for tuning in today and I'm thinking about my to-do list now. Thank you for tuning in today. And thank you so much to Dr. Nixon for giving an excellent talk on the use of endocrinology in conservation, and especially revolving around bats. It is a field that is ever growing and is only going to be more useful as we start monitoring our populations with different methods, especially ways that help answer some of these more detailed questions about how our anthropogenic world is affecting our wildlife on a physiological scale. So thank you so much, Lydia. And again, if you want to tune back in to see the talk again, please check back on our YouTube channel for GBATnet within the next couple of days.